Thomas. I'm a PhD student in Catherine Lennon's lab at the University of Kentucky. And today I'm going to tell you a story about how I came to be where I am, quite literally. So unlike most students in my graduate program, I am a Kentucky native, and 2 years ago I moved from Louisville to Lexington, which you can see here, I think, Louisville to Lexington for grad school. And while the distance between where I was and where I am now was only about 80 miles, which is well within my cruising range, this shift in my host has caused some isolation from my friends and family in Louisville when I moved to Lexington. And the most obvious response to this move has been my habitat. So clearly, if I'm not in the same physical location as my friends, it becomes a barrier to our relationships. But not only has our habitat changed to not overlap anymore, there's also been other reactions to this shift. So now that we no longer share the same host or workplace, our schedules don't overlap. We can't just go out and grab a beer after work because our hours are no longer the same. We've also had some breakdown in communication. Since my move, I used to just be able to pop across the hall and chat with my friends, and now it takes more effort to text or call or email. So the fact that I have experienced 3 forms of essentially premating barriers from just a relatively short move across the state got me thinking about some of the classic thinking about how host shifts drive speciation through divergent selection on habitat preference, resulting in reproductive isolation. And there are innumerable forms of premating barriers, but my research is going to focus on these 4. Organisms must be in the right place at the right time, be able to recognize each other, and morphologically fit together. If any one of these potential hurdles are not met, some sort of reproductive barrier will arise. At each of these stages, there's an opportunity for divergent selection on hosts to impact isolation. For example, habitat isolation might result if animals mate on the host and the host has changed. Temporal isolation might result if the host shift results in some sort of circadian disruption or a seasonal difference based on host. Chemical communication might be impeded if the animal incorporates host properties into its pheromones or the new host is aversive to animals on the former host. And size-assortative mating could result in mechanical isolation when a new host might promote or restrict growth and resulting in morphologic variation. Despite all of these opportunities for divergent selection to have significant direct or indirect effects, until recently, the thinking in the field has considered habitat isolation the most important or most direct barrier resulting from a host shift. So I got curious about actually testing this and testing other potential mechanistic links between divergent selection and reproductive isolation, and ultimately the impacts on speciation. Studying barriers to reproduction is most effective in a system in the middle of this speciation continuum, for which the speciation process is incomplete. So this brings me to my study system of Neodybryon pine sawflies. Pine sawflies are a group of hymenopterans specialized on pine trees, and like most plant-feeding insects, pine sawflies are intimately associated with their host plant at all life stages. Adult females call for mates on their preferred host. The males respond to her call, and the pair mate on the tree. The females then dig these little pockets inside pine needles, which is where she will lay her eggs. The larvae hatch out of those eggs and then consume the pine needles. And then they pupate in cocoons either on the pine needles themselves or in the soil directly beneath the tree. And then adults obviously emerge to repeat this cycle. Specifically, I'm using a pair of sister species of Neodybryon sawflies to address these questions about premating barriers related to a host shift that these guys have experienced. The top image is Neodybryon lecontii, who's as a generalist, the species that uses more robust, thicker needled pines and avoids white pine. The bottom image is Neodybryon pinetum, which exclusively uses white pine. In the center, we have morphologic hybrids, which are this kind of cream color instead of the yellow or white of the parental species, and then they have that rusty brown head cap instead of red or black. On the far right, we have an admixture plot that confirms these hybrids genetically. And additionally, we have multiple lines of evidence that these species experience divergent selection due to a host shift. First, this phylogeny shows a repeated pattern of host shifts associated with speciation events. Second, individuals found on the same host are less genetically diverged than those found on different hosts, a classic signature of isolation by environment. And third, significant extrinsic post-zygotic isolation based on host has been demonstrated via egg hatching success on preferred and non-preferred hosts. So my question now is, how has this divergent selection on host contributed to different premating barriers? And while I'm working on all 4 of these different premating barriers, today I'm just going to tell you specifically about my projects concerning habitat and mechanical isolation and how they might relate to a host shift. So we know that females of these species differ in host preferences and that these preferences are under divergent selection because females who lay eggs in the wrong host do really poorly. So they cut their pockets too deep, they dry out the needle which dries out their eggs. Those guys aren't going to survive. But does this female preference translate to habitat isolation? So to test for male host preferences, we tossed a male into a cage, a mesh cage with a white pine and a non-white pine, kind of just to see where they spend their most time. And whereas females have a very clear preference for host, males don't spend any more or less time on the host of that the females of their species prefer. And in fact, Neodybrion lecontii spends most of his time just wandering around on the nest cage without making a commitment to either host. So this data led me to my first big question: do males use host cues in mate searching at all? And one way to go about answering this question is simply to go out in the field and see where these dudes are hanging out. So I placed sticky traps baited with common synthetic pheromone lures in white pines and non-white pines around Lexington and collected males at regular intervals through the summer. And if males are using host cues, I predict that males will be found in greater abundances on the host preferred by their conspecific females. So for Pinetum, we would expect to catch more Pinetum males on white pine than on Virginia pine or other non-white pines. And for lecontii, it's just going to be the exact opposite, where they should be hanging out more where their females are going to be hanging out. And in the summer of 2018, I collected approximately 6,500 individuals, but unfortunately, they can't be identified to species morphologically. So for now, all I have is some comparative abundances based on host, but I think I can see a few patterns based on this limited information. First, both hosts seem to follow this pattern of 3 large peaks, which represents the 3 generations we typically see in Kentucky. Second, traps on white pines, so the black line, always had more individuals in the traps than non-white pines. And third, this red line that kind of starts right in the middle of the trapping season here was kind of a happy accident when an angry neighbor got mad and he didn't like the way the trap looked in the public park. So I took it and shoved it in a bush next door just to keep the site going. And it still collected males in the same pattern as their preferred host, which I thought was interesting. It's at reduced quantities, but it's still the same pattern, and they're still catching dudes in a poplar tree. So far, there's some mixed evidence for habitat isolation between these 2 species. There are differences in abundances on the different hosts, but since I can't ID them, I don't know if this is reflective of species preference yet. So I'm currently working on identifying these individuals using a PCR-based diagnostic assay to answer this question and get at this a little bit more. And in contrast, the traps in non-pines are still attracting males, which is pretty interesting. This could mean that the males aren't using host at all to find mates. It could reflect a combined effect of host and pheromones, so there's some sort of synergistic interaction, or it could be just that the lures that I'm using are so strong, it's overwhelming these guys' little brains, and they just go wherever they think this huge blood of females is. So to account for this possibility, I'm working on extracting host volatiles to use in electroantennogram and olfactometry assays to examine the physiologic response without the impact of pheromones, so stay tuned on that. But even if we help these guys out a little bit in the lab, we put them in the same place at the same time, there's still something else keeping them apart from mating at the same rate as they do with conspecifics. And this figure shows a reduced willingness to mate with heterospecific individuals in both directions. So clearly there are other barriers in play, even once those initial hurdles have been cleared. So I'm working on all of these, but today I'm going to tell you a little bit about the work I've been doing examining the potential role of genitalia variation between species and how it might contribute to premating isolation. So we know that there are differences in female ovipositor morphology, egg-laying pattern, egg spacing pattern, and that these traits are divergently selected based on host. In this figure, females with larger, more lecontii-like ovipositors had a significantly reduced hatching success because when laying on the white pine, because these are the guys that are going to butcher the needles, cause the needle to dry out, and kill all their eggs, causing them to dry out, die, sad face, no good. So my next big question is, are there correlated traits in male genitalia? And since insect genitalia are incredibly complex, I chose to start by focusing on non-intermittent organs, specifically clasping organs called harps, these little guys right here, because I think they contribute to the tight coupling, the tight fit that we see during mating. When these guys are copulating, we've observed the females walking around, sometimes even flying around with the male still really attached to her abdomen. So clearly there's something going on with that really tight fit. And so then I got interested and took these scanning electron microscope images, and we can kind of see these harps extruding out from the male's abdomen and really clasping onto the female, almost like a suction cup. So I can imagine that a less than perfect fit is going to result in an unstable pairing, shorter coupling time, and potentially reduced sperm transfer. So to investigate this, I started by using imaging software, and I just measured the area of these regions. And since one way to get smaller genitalia is simply to get a smaller body, I checked for body size variation using wing length as a proxy measurement, and Lecantii and Pineta males are not significantly different in wing length. But Pineta males do have significantly smaller harps, which correspond with the smaller ovipositors that the females of that species possess. So again, results for mechanical isolation are mixed. On the one hand, I have demonstrated significant variation in at least one aspect of male genitalia between species. However, I don't know if this matters for mating success. To address this, I plan to look for size assortative mating within and between species based on size and copulation success. I can also generate hybrid populations, which will have a wide variety of parental traits, and I can look for mating success or failure based on genital size. For example, males with the larger, more Lecantii-like genitalia might be more likely to be successful with Lecantii females and vice versa. So stay tuned for that too. So I hope this story has convinced you that while historical thinking of host shifts and speciation postulate that habitat isolation is the barrier resulting most directly from divergent selection on hosts, there are many other forms of premating barriers that are influenced by a host shift, and this warrants some additional future studies. And with that, I think I have a few minutes for questions. Thank you to my lab and all of our funding sources. Yeah?
So are the carps and the ovipositor developmentally the same in males?
Yeah, we don't know. That's a really good question. So yeah, it would be interesting to see, are they the same genes kind of controlling those? And it's actually kind of neat. Another student in my lab is doing a QTL mapping study on ovipositor size and other egg-laying behaviors. And so I could potentially do the same thing with the male genitalia and see if they're the same genes kind of working on that trait. Yes?
Are there known sex pheromones in these species of sawflies?
Yes, there are known sex pheromones. The major components in the two species are that seem to be the same. So there's probably some difference in abundance in the less abundant compounds of the pheromones. So we've got some collaborators in Europe that are working on the pheromone aspect. I'm starting to work on cuticular hydrocarbons and profiling those to see if they're different. We could also kind of do a you are what you eat study to split the colonies. If you raise them on different hosts, does that change their pheromones or CHC compounds? So we're getting there. Yes?
Rather than using pheromone traps, have you thought about using traps baited with volatiles from the two host plants?
So place host volatiles in the wrong host?
Exactly.
Haven't thought about that, but that's a really cool idea. Place white pine volatiles in a poplar tree or something. Yeah, thank you for that. I'll give that a try. All right.